Welcome to Structural Biology Communications interviews with authors. I am John Aguirre from the University of York in England, and connecting from Universidade de Sao Paulo is Professor Cryst. Today we are welcoming Shenkutie from Hawassa University in Ethiopia and John Hughes from Justus Liebig University Gießen in Germany to discuss their article Expression, Purification and Crystallization of the Photosensory Module of Phytochrome B from Sorghum bicolor. Sinta, John, it's a really big pleasure to have you here. So could you perhaps provide a brief overview of your research paper in lay terms for our audience?
Well, thing is, plants are regulated very closely by phytochrome. It's a photoreceptor that we work on. It's a light sensor, and it controls almost everything about how the plant grows, how it germinates, when it flowers, and so forth. And we'd like to find out how this photoreceptor, how this photosensor works. This kind of photoreceptor is called phytochrome. It exists in bacteria as well as plants, but animals don't have this thing at all. And we want to know how it works. And we've been working on how the molecule works in bacterial phytochromes for many years. But this paper that we published describes how we managed to start working on plant phytochromes and using E. coli to produce them and purify them. And that's really what Sinta's PhD thesis was all about.
Yeah, great. Very interesting work, I have to say. Could you share some of your major challenges or obstacles that you encountered during the research process? How did you overcome these challenges, and what lessons did you learn from them?
So some of the challenges that we encountered during this research project includes, like as John told you, these are plant proteins, and if we want to extract these proteins from plants, we need probably tons and tons of sorghum. So the solution for this is to use recombinant production in E. coli. But when you produce recombinant proteins in E. coli, there comes another challenge. That challenge is our protein is relatively large. Even if we are not working with the full, even if we are working with part of the molecule, that's why we call it the photosensory module. If that one is still bigger. And another thing is this protein has a cofactor. So because it serves for plants to detect light, like, you know, in humans, in our eyes, there is a rhodopsin that is a cofactor. In plant leaves, these phytochromes have another bilin cofactor, which serves the same purpose like rhodopsin. So this one is not expressed in E. coli. So we have to make or engineer the E. coli to produce these plant chromophores. And once this has already been established at the Hughes lab, and this makes life very easy. So another thing probably I should mention as a challenge is because these proteins are light sensitive, okay, most of the experimental setup, like the purification, the crystallization, and even the diffraction experiment has to be done in the dark. So let me simply describe it, how we fish it. We go to the dark room at 4 degrees centigrade in the cold room, and we use the safe light in our head, and we have to do everything for hours and hours in cold room in the dark. That was very challenging.
Very interesting. What we do for science, right?
Yeah.
Exactly. What were the yields like? How much protein were you able to get typically?
After we optimized the production condition, we were lucky. We were able to produce large quantities of holophytochromes in E. coli. Probably, I think at one point we produced 0.1 gram of protein from not just probably from 16 liters of culture.
Yeah,
it doesn't sound like much, but it's a hell of a lot for the sorts of molecules that we're working with.
Yeah, exactly. Right. How does this study link to your wider vision of science, and what is next in this line of research?
Well, I've always wanted to work on something to do with physics, but unfortunately, my mathematics was never good enough, so I became a biologist instead. And I always wanted to do stuff with astronomy, but yeah. So I ended up being a biologist working on light. And I found plants much more interesting than animals in physiology because I don't like the idea of cutting up little animals with a knife. And I started working on phytochrome, and I've been working on it now for nearly 50 years. So photosynthesis, of course, is in a way more central, yeah, to our world, to human condition. But everybody works on photosynthesis. So I thought, actually, end of the 70s, when I was still a student, that phytochrome would be a little bit more interesting, yeah, a bit less competition, perhaps. And I've been working on it ever since.
What do you think what comes next in this line of research?
Well, there are different things. I mean, the research isn't finished yet, yeah? We're finding out how the molecule works, like, you know, like in some detail, like how the photons are absorbed and then how it communicates the information to the cell. But of course, it depends what you mean now, because I mean, we want to say, okay, in agriculture, there are very many things that you can do with the help of phytochrome manipulation. I could talk a bit more about that if you're interested.
Yes, please.
But yeah, okay. The other thing is the basic science that phytochrome is a photoreceptor, and it does very interesting things. And those things might be very useful for all kinds of stuff, you know, that might be to do with development of light-emitting diodes or solar panels, or at the other end, even research into how the brain works. And in fact, all of those examples already exist. It's not just me making jokes about it. Yeah? So there are quite a few things that could come out of it. As far as agriculture goes, if you want that in particular, these days, farmers plant crops extremely close together. So a very, very, very high density cultivation of crop plants. And that means that these plants are in each other's shade. And one of the things that phytochrome does is allows plants to detect shade. So what happens is the plants are trying to avoid the competition by growing higher and taller and taller and taller, all at the same time. So they all lose. Yeah, not one can outgrow the other. They all lose just trying to get taller and taller, using up the energy that they have to grow taller instead of making grain. So one of the things that people are working on now is to produce plants that don't do that anymore. And it's not so simple because you can't just get rid of the phytochrome. You have to manipulate how it works. And people are interested in making aviation fluid from seed, from plants that have been manipulated to do this. And in fact, I gather that Exxon and some other huge petrochemical companies are putting a lot of money in this research at the moment. So there's a huge variety of stuff that can be attached to the sorts of things we do. But of course, the problem is the very basic science, you have to find it out before you can say what it's going to be useful for. So it's a gamble. Yeah. But sometimes it pays off.
Thank you.
Yes, I think you sort of start answering my next question is how do you envision your research impacting the field or society at large?
Yeah, I think one has to be a bit careful about knowing in advance with basic science where it's going to go. Companies like Exxon and IBM spend a lot of money on basic research. And they don't do it just because, you know, to save money. They do it to spend money. So it's a very difficult question to answer. And if anybody could answer that question, then of course we'd save a huge amount of money. But that's not the way it is. Yeah. So I think the fact is that one tries to do fundamental research as well as one can, and you keep your eyes open for the possibility of it being used, or the information being used in a different context, yeah, or maybe in a similar context, you know, depending upon which what you're talking about agriculture was one aspect, but photoreceptors like rhodopsin and phytochrome, they have very interesting properties in the living cell, and some of those you can use in very different contexts than they would be in this case, the plant. Yeah, so I mentioned brain function. There's another plant photoreceptor that's being used very extensively worldwide for the past 5 years or so to control nerve, let's say, brain function. Yeah, you can just activate the photoreceptor and the mouse starts running. And you switch off the lamp, the LED that activating the photoreceptor, and the mouse stops running. It's all been done. Yeah, so lots of things can be done, but it's hard to know in advance what's going to work, right? And if you can improve agriculture with it, or maybe better, you know, make a better solar cell, or find out how the brain works, well, not such a bad thing.
Yeah, basic sciences is still very important, right? Yeah.
Right. Our next question has been answered by John, I think. However, I would like to get Sinta's thoughts on this. Why is this study important to you? Or in other words, not your funder or institute?
I see. So personally, yeah.
Yes.
So I think I'm going to tell you a true story. When I was undergraduate student, probably some 20 years ago, I have my first encounter with X-ray fiber diffraction image, the famous photo51 in molecular genetics class. And then my professor at that time, he asserted that this X-shaped diffraction image unequivocally proved that the DNA is double helix. So it's a proof for double helical structure. And then after the class ended, I was pondering for hours and hours after the class, just watching the image and then questioning how is this X-shaped image translate into helical structure. So I was looking at it from different direction and it just doesn't make sense. I can't translate it. But now, after many years of exposure to structural biology, I have a deeper understanding of concepts, important concepts like real space, reciprocal space, Fourier transformation, and those things. And I can connect the dots. It's a long journey, but I think personally, scientifically, for the guy from Addis Ababa University who happened to go to synchrotrons and observe how atomic structures are determined and understand this process is scientifically immensely fulfilling experience for me.
Excellent. Thank you.
What motivates you guys to pursue this particular research topic?
Well, so the particular research on phytochrome, yeah. I remember, like Sinta was just saying, when you're young, yeah, you hear things, they have big effects, and most of the things you hear, you just don't even bother to take attention of, right? When you're that age. So I heard a talk from a professor in Queen Mary College in London when I was an undergraduate in 1976 about phytochrome and how it controls seed germination and why that's a quite important environmental function, sort of light-dependent seed germination. And it seemed to me like a nice round subject. I'd never really, I think I'd heard of it, but wasn't part of our course really. And I thought that would be something that would be worth looking at if you're interested in some physics, some light, physiology and so forth. And I sort of got a little bit more interested, a little bit more involved. And then slowly, you know, you get to do a PhD, and then you get a grant to visit Germany. And I stayed in Germany now for the last 40 years. And you know, if you're lucky, you make some discoveries, somebody give you a nice job, and now I retired a year ago. So all worked out very well. Lucky.
He definitely did. What advice would you give to aspiring researchers based on your experience?
Well, Sinta, you have a go. I have an opinion as well. Sinta has one.
I think you go ahead. First, our audience will learn from the PI and then from the student.
Sometimes the PI learns from the student too.
Yes.
So my advice, yeah, this is wisdom from aging white males. Yeah, you have to be careful. I think what people should do, yeah, is to find something quite specific to work on, not just general things like I like plants or something, you know. You want to something a little bit more focused than that, and something you find interesting. Because it won't always work in the lab. You'll have periods when it's actually rather depressing, but you have to push through. Otherwise, you might as well stop from the start and go to work for a bank. Yeah, you stick to it, but most important, you keep your eyes open. Whoops, and you look to the left and you look to the right while you're going forward, so that you keep in touch with what might be useful from another approach, a method, or whatever. And what I also think is very important to keep in mind is the capitalism of research, meaning that unless you're the best in your field, you should always try and avoid competition. That's what all capitalists always try to do, is to avoid competition so they have a monopoly. That means they can make more money for less work. And that's not bad in science either.
Thank you. That was a very interesting advice. So, Sintayehu?
Okay, yeah, thank you. As a PhD student, I have learned a lot of lessons, and for other PhD students or beginning researchers, I have a couple of advice. The first one is about the value of documentation. When you are working in the lab, make sure you document each and everything that you are working, the good, the bad, and the ugly. When your proteins are expressing happily, you document it. When you don't see anything, when your proteins doesn't express, you document it. When you break glass slides, when you break equipment, also you document it. This is also the ugly, but this is part of the science. The second thing is the value of collaboration. As a researcher, when you are working in the lab, you are not alone, and you can't do everything on your own. So be free to ask for help, support from your PI, from other PhD students, from postdocs. So collaboration and active discussion is very important. And probably I don't know what else can I say. I think this is it. These two are very important.
Yeah.
I can think of much worse things to conclude a video with. I mean, that's very sound advice.
Yeah. One thing, I add one thing to that, which is actually quite interesting, because I had this discussion with some people recently. There's no such thing as incorrect data. It cannot exist. All that happens is that people misinterpret it. So if you have a measuring something, you don't even bother to switch on the machine. Yeah, it produces a lot of zeros. The data is perfectly correct, just that you have to remember that the machine wasn't switched on. Yeah, because you think of how much science has got wasted because people thought they understood it, but were completely thinking the wrong way, you know, probably for thousands of years. Yeah, and only sometimes people realize, no, this is wrong. You have to interpret it this way, and then everything makes sense again.
Absolutely. Right. I think we've come to the end of the interview. Thank you very much, Sinta and John.
You're welcome. No?
Best of luck for the future.
Okay.
Thank you.